Now, last week we had our session. We did discuss quite a number of things. We were looking at the DNA. We are trying to understand the structure and the function of the gene. And maybe just a recap of what we discussed. We were able to look at a chromosome as a structure that contains a DNA molecule. Now, and each DNA molecule is divided into segments or into portions. Each of those portions or segments represents a gene. And we were also trying to look at the role of genes. And it was very clear that there are three important roles of the DNA. One translates the genetic information into characteristics of an organism through protein synthesis. We were able to look at what happens during DNA replication. And today we are going to even watch a video that will be showing how this process takes place. We are also discussing protein synthesis, and the proteins synthesized determines the characteristic of the organism. So that was just a recap, and I want now to get into today's business right away. I want to share my screen. Okay, now on your screen is one of the items that we discussed, and I would like to ask one of the students to tell us what is it, or what we can, or what we are able to see on the screen. We have Siham Mohammed. Can you hear me? Siham Mohammed, can you hear me?
Yes, Molim, I'm able to hear you.
I'm asking a question to Siham Mohammed.
Yes, Siam can go on.
I can hear.
Proceed. Yes, you can hear me. Now I'm asking, what is it that we are seeing on the screen? What are you seeing on your screen? Yes?
Unzipping of the chromosome.
The unzipping of the DNA molecule?
Yes.
And the formation of? Yes? Okay, you can see from one DNA strand, we have two DNA molecules that have been produced. From one DNA molecule, we have two DNA molecules. And what you can see is that these two DNA molecules, this one and the other one, are exactly replica of each other. And both of them look at, look the same as the parent or the original DNA strand. Now, when we say that the role of DNA is to store genetic information unchanged and transfer the same from one cell to another and determine the characteristic in an organism. So this genetic information, which is stored in the DNA molecule, it is replicated when the cells are reproducing or when the cells are forming new cells, and therefore that coded information is transferred unchanged. So we have the template strand. Of course, you can remember that. Can remember this basically showing the process of replication. We did discuss this. So that is still replication. So I have a video here that I would want us to watch. It's about DNA replication.
Cells like these prokaryotic E. coli cells replicate themselves quickly and efficiently. Part of the process of asexual reproduction is the ability of cells to make identical copies of their DNA before cell division occurs. Prokaryotic cells that reproduce by binary fission rely on the fast, accurate process of DNA replication to ensure future generations of cells will have the same genetic instructions as the parent cell. The structure of DNA aids in the speed and accuracy of replication. Double-stranded DNA is a polymer of 2 strands of nucleotides which are hydrogen bonded to each other to form a double helix. Nucleotides are molecules that consist of a deoxyribose sugar, a phosphate, and one of 4 nitrogenous bases. The phosphodiester backbones consist of alternating sugar and phosphate groups. The nitrogenous bases include cytosine, thymine, adenine, and guanine. Cytosine forms 3 hydrogen bonds with guanine, and thymine forms 2 hydrogen bonds with adenine. This is referred to as complementary base pairing. The double helix will have one strand oriented in a 5' to 3' direction relative to the hydroxyl group of the deoxyribose sugar, and the other strand oriented in a 3' to 5' direction. This shows the anti-parallel nature of the DNA strands. The complementary base pairing in the structure of DNA allows replication to be executed in a semi-conservative manner. Each strand of the DNA molecule is used as a template in the creation of a new double strand. Replication begins with double-stranded DNA being separated, and each original strand, called a parent strand, is used as a template for the complementary base pairing of nucleotides to make 2 new molecules. DNA replication occurs in the 5' to 3' direction, adding new nucleotides to the 3' end of the newly forming strand. DNA replication will begin at a specific area of the molecule called the origin of replication. The origin of replication denotes the area of active replication called the replication fork. In order to understand how complex eukaryotic organisms replicate DNA, scientists first studied replication in prokaryotic models like E. coli. A number of enzymes are needed for replication to proceed once the replication fork is established. Helicase separates the strands of the double helix, and single-stranded binding proteins stabilize the newly single-stranded regions. DNA gyrase is used to make sure the double-stranded areas outside of the replication fork do not supercoil. Once the replication fork is stable, DNA polymerase catalyzes the addition of new nucleotides to the growing daughter strand. Other proteins, such as beta clamps and the clamp loader, help hold the DNA polymerase in place on the DNA. Short sequence.
Okay, from what we have managed to watch, I think it's quite clear what happens.
Cells like these prokaryotic E. coli cells replicate themselves quickly and efficiently. Part of the process of asexual reproduction is the ability of cells to make identical copies of their DNA.
Okay, so from the short video we have watched, I think it's quite clear how DNA replication takes place. And that is precisely what we did cover in our previous lesson. We are now going to look at what role does the DNA play in protein synthesis. Remember, from our very last lesson, we said the proteins synthesized in an organism are controlled by the DNA. In other words, the genes are the ones which determines which proteins will be synthesized. And the protein synthesized gives the organism the characteristic. Now, I want to give an example at this point in time, although I may have given this in our previous lesson. Now, you happen to have a gene in your skin, and in particular, a Malpighian layer of your skin has what we call melanocytes. So within those melanocytes, those are the cells that produce melanin. They are genes, or there's a gene which is responsible for synthesis of melanin. So what therefore means is that when you have the normal gene which is responsible for synthesis of melanin, melanin as a protein is synthesized. And presence of melanin in your skin gives you a characteristic. You have normal skin color. So what happens to those individuals who may have a gene which is unable to code for synthesis of normal normal melanin? They produce a protein which is not melanin, something different, which then gives them a characteristic that we call albinism. So, what I'm saying is that as we move on, we need to understand what happens when proteins are being synthesized. What role does DNA play? When I talk about DNA, I expect that what is supposed to come into your mind, we are talking about a gene. And I'm not saying that DNA is the same as a gene. So what I'm saying, and I want to repeat, a chromosome contains one DNA molecule. Then that one DNA molecule is divided into segments, and each segment represents a gene. Each segment represents a gene. So as we talk about the role of DNA, now in general, we have genes within the chromosome. How do these genes bring about protein synthesis? That is precisely what we want to discuss. Okay. Okay, so let's move on to the next slide. Okay, now what happens is that the information which is stored in the DNA, in the DNA molecule, is copied and transferred to the cytoplasm. Now, what happens? I want to explain that. What happens is that, let's look at this example here. Now, we have a cell. And within a cell, we have a nucleus. Now, within the nucleus, this is where we have the chromosomes. We know from our basics that the structures which are responsible for protein synthesis are located within the cytoplasm. Now, what do we call these structures that are responsible for protein synthesis? Somebody with an idea? We refer to them as ribosomes. We refer to them as ribosomes. Now, this DNA coded information within the nucleus and within the chromosomes. How does it reach the ribosomes in the cytoplasm? So that the ribosomes can be able to synthesize the required protein, which will give the characteristic to the cell or to the organ cell. So it means, and of course, remember that, okay, although maybe you are not aware, DNA molecules do not move out of the cytoplasm, out of the nucleus. Chromosomes are always within the nucleus. They don't move out. So it means, therefore, there is an intermediate. There is a communication between the ribosomes and the chromosomes in the nucleus, which carries the DNA. So what therefore happens is that we have what we call messenger RNA. Now, the messenger RNA is another nucleotide, is another nucleic acid, which is involved in carrying the genetic information from the nucleus to the cytoplasm and specifically to the ribosomes. Okay? So the messenger RNA is what I want us to talk about. Now, this messenger RNA, I need to clear this so that we can discuss more. So I'm just sharing my screen. At least you can see something. What I've just explained. Okay, now I want to explain something. And what happens is that I'm just doing a small portion of the DNA. So if this is guanine, this is cytosine. If this is thiamine, this is adenine. If this is adenine, of course, this is thiamine. If this is cytosine, this is guanine. Okay, you know the base pairing, how they pair. Okay, so what happens is that we know when proteins are being synthesized, proteins are made up of building units called amino acids. Maybe a recap from what we covered in Form 1 under chemicals of life. If you remember, the building units of proteins are amino acids. So what happens is that when proteins are being synthesized, amino acids are held together, or they combine through a process we call condensation to form more complex protein molecules. And these protein molecules, they are made up of what we call peptide chains. So what happens as far as the DNA and the protein synthesis is concerned? In my case here, I've drawn a short portion of, okay, let me draw something else here. Okay, I've drawn the nucleus and a small portion of the chromosome containing one particular gene. And that gene is now the one that I have drawn out here. Nataka kuongeza hii kidogo so that we can appreciate. So if this is guanine, that is cytosine. If this is guanine, again, cytosine. If that is thymine, adenine. If this is cytosine, this is guanine. Now, assuming that what I have drawn here is a small portion of the DNA molecule and representing a gene. We have a gene in this example with 1, 2, 3, 4, 5, 6, 7, 8. I can even under the 9 one. Maybe that is thymine, this is adenine. So I have a small portion representing a gene. DNA is a wrong structure, is a wrong polypeptide chain. I mean, it's a wrong chain. But now I'm just narrowing down to only a small portion of it, which is representing a gene. Now, supposing this is gene A, which is responsible for synthesis of melanin. Okay. Now, the arrangement of amino acids in what we call polypeptide chain. Maybe that is one amino acid. This is another amino acid molecule. This is another molecule of amino acid. You know, there are different types of amino acid molecules. In nature, we have 20 different types of amino acids. 10 of them essential, 10 non-essential. You know the reason. So one, okay, let me see the codons I have here. One, I have, okay, there are 3. Now let's assume that the gene responsible for synthesis of melanin consists of the nucleotide molecules that I have drawn here. In total, there are 18 nucleotide molecules. Now, for the arrangement of polypeptide, for the amino acids in a polypeptide chain, it is determined by the base sequence along the gene. Now, let's assume that the first amino acid here, the codon responsible for positioning of this particular amino acid on that particular position is, this is GGT. So we have the first codon, GGT. That is the first codon. Then we have the second codon is, what is it? This and thymine. Then we have the third one. The third one is adenine, cytosine, thymine. Adenine, cytosine, thymine. Now, what it means is that this information which is stored in this DNA molecule, I hope you can see that. Assuming for this DNA, for this amino acid to be positioned here, the codon which brings it there is this the codon. We have the other codon for the other amino acid and the other codon for the other amino acid. So what happens is that for the ribosomes to be able to synthesize this particular molecule, I'm assuming this is one molecule of a protein called melanin. This is what I'm assuming, that a molecule of melanin consists of 3 amino acid molecules. Now, what therefore happens is that for protein synthesis to take place, the positioning of amino acids along the polypeptide chain is determined by triplet bases. Okay, so like the example I've given here, if this is gene A responsible for synthesis of melanin, this coded information will be copied on a messenger RNA, and it will be transferred to the ribosomes in the cytoplasm, who will be able to interpret this coded information and synthesize melanin. Maybe if you look at what we have in the next slide. I need to clear this so that at least you can be able to see. Now, every set of 3 bases along the DNA strand is responsible for bringing into position a particular amino acid of a polypeptide chain. Basically, that is what I was trying to explain. Now, we have a simple example here. The base triplet, we call it a codon. And it is meant, or it's supposed to code for a particular amino acid of a protein molecule. So we have examples of such DNA codons. We have AAA. This is adenine, adenine, adenine. They code for an amino acid called phenylalanine. If you take this as one of the examples of amino acid, then we have triple thiamine, TTT, coding for racine. Racine is another amino acid molecule. Then CAA, cytosine adenine, adenine, code for valine. Valine is another amino acid molecule. CTA, coding for as the positioning of aspartic acid. Now, supposing we have a DNA consisting of this codon, AAA, TTT, going down. Maybe I can let me take my pen again. Now, supposing we have this codon, A, and then we have this one following here. Okay, then we have this following here, and then we have this down there. Okay, now we then assume that this is one gene. So what happens is that now in a protein molecule, the first amino acid molecule will be phenylalanine. Assuming that is phenylalanine, it will come first, and this will be linked to racine. Let's assume that this is racine, and then we have valine. Let's assume that that is valine, and then we have the aspartic acid. Let's assume that this is the aspartic acid. So we have now, this is a protein, one molecule of a protein. This one molecule of a protein consisting of the 4 amino acid molecules. And you can see for these 4 amino acid molecules to be in their particular positions along this polypeptide chain, it has been determined by the coded information which is in the gene. So this coded information in the gene, how does it reach the ribosomes in the cytoplasm so that they can interpret that coded information and synthesize this particular protein? Okay, so another point I also want you to appreciate is that these 5 different amino acids, they can make different types of proteins. What happens is a matter of changing the positions of the amino acids in the polypeptide chain. So any questions so far? Questions so far? Yes, protein synthesis take place in ribosomes. Where are ribosomes? They are found in the cytoplasm. So DNA molecules are confined to the cell nucleus. Okay? Now, you need to understand this. If, for example, we are talking about the human body or a cell in your body, which has 46 chromosomes, it means that cell has 46 DNA molecules because a chromosome carries one DNA molecule. Now, those DNA molecules are confined within the nucleus of the cell. So the question is, that coded information, which is confined in the nucleus of the cell, how does it get into the cytoplasm where the ribosomes are located? That is the question. And the answer is, we have an intermediate, we have a means of communication. And that's what we are going to discuss, what we call the messenger RNA. We call it messenger RNA. So the cell has got special molecule that mediates between DNA molecule. Okay, of course, you can say what is on your screen, what we call the messenger RNA. So messenger RNA, as I said, it's another nucleic acid. Okay, and there are two very important things that we need to understand about it. One, RNA is a single strand. It is not a double strand like DNA. And it is made up of units called ribonucleotides. Okay, DNA is made up of nucleotide molecules, but this one, we call those molecules, they are called ribonucleotides. And in this RNA, the base thymine is replaced by the base uracil. So where we have adenine, it will not pair. Okay, we don't have thymine in a messenger RNA. Messenger RNA, we are saying it is just a single strand. It's just a single strand with organic bases. So if you remember, allow me to use my simple diagram to illustrate that. I'm trying to put something on the backbone, of the messenger RNA. Now, this straight line, I have put some structures on it. I have put ribose sugar instead of pentose sugar. It is ribose sugar. This is ribose sugar phosphate group. Then this is an organic base. This organic base could be uracil. This is phosphate group, ribose sugar, an organic base. So a molecule of RNA consists of, maybe I can use a different color for you to see. We have, this is ribonucleotide molecule consisting of a ribose sugar, phosphate group, and an organic base. So the backbone therefore consists of ribose sugar alternating with a phosphate group. Then we have organic bases sticking out at some regular intervals. So it is a single strand. This is one nucleotide molecule. This is another nucleotide molecule. So they form one single strand, which we refer to as messenger RNA. So I beg to move on if there are no questions. So maybe we can have a short look at this video. We learn more about DNA in the role of DNA in protein synthesis.
The role of deoxyribonucleic acid or DNA in protein synthesis is that of a blueprint. It is a guide to the structure of the proteins being produced. Without DNA, the ribosomes in any given cell would not know what order to put amino acids in. DNA has the same function in both prokaryotic and eukaryotic cells, although there are subtle differences. DNA is a chain of nucleic acids arranged into two polymers or strands. Each strand has one set of amino acids that connects to an opposite amino acid on the other polymer to produce a structure that looks like a window cleaner's ladder. The order of amino acids is a genetic map of information that tells the cell how it is to be structured and tells the cells how to combine to form a larger organism. The information is used directly to build cell components such as ribonucleic acid, RNA, and protein. The presence of DNA in protein synthesis is vital. Protein synthesis is the act of creating a new protein within a cell. The entire process takes place within a ribosome, a kind of protein factory, within a cell. Free ribosomes in eukaryotic cells and all ribosomes in prokaryotic cells synthesize proteins in the cytoplasm. There are many steps to the protein synthesis process. The use of DNA during protein synthesis takes place in the first stage called amino acid synthesis. The second stage is called transcription, and the final phase is where the ribosome translates the information into protein. A protein called helicase splits apart both polymers of DNA in protein synthesis. One of the strands will contain the protein blueprint the cell requires. This strand will be copied into messenger RNA, mRNA, when the mRNA is organized so that it is made up of the opposite amino acids to those present in the DNA section being copied. The mRNA then takes the information to the ribosome. The ribosome will process the mRNA so that it translates the amino acid code using the opposites of those in the mRNA, therefore returning the chain back to its original form. From this, the ribosome makes proteins. Organisms are not capable of synthesizing all amino acids. There are around 20 known amino acids in the world, and humans can synthesize around 12 of them. The rest are ingested through food and sometimes drink. The prokaryotic cell will convert DNA in protein synthesis directly into mRNA. Eukaryotic cells, however, first transcribe the DNA into heterophil nuclear RNA, NA. This NA is created when the polymer section is capped with 7-methylguanosine and a poly-A tail. The cell then converts NA into mRNA.
Okay, so I hope you have learned something from that short video. Now, from our video and from what I had discussed earlier, I think it's possible to give the differences between DNA and RNA. So you can see one of the difference is the type of sugar we have. Like in DNA, it is the oxyribose sugar. We have both in RNA. Then we have the two nucleotide chains. So one is a single strand, that is RNA, is a single strand, and the other one is a double strand. Then DNA, you can see thiamine instead of uracil. For the other one, we have uracil instead of thiamine. The role of DNA in protein synthesis, you can see what happens. We have heard about transcription and translation. Okay, there are three terms here. There is replication, transcription, and translation. Now, replication, we already discussed. It's a process through which a DNA molecule produces an exact replica of itself. Transcription is now the process through which a DNA produces a single strand of messenger RNA, where the DNA strand acts as a template. So what happens during transcription is what I'm going to discuss now. And later on, what we call translation is now.
Munene.
Yes.
Yes, student named Bahja Mukhtar is asking if you can repeat the page of differences. She did not get it clear.
The differences.
Yes.
Okay. So the differences is about the sugar in each one of them. The other one is the number of strands. One is double strand, the other one is single strand. And the other difference is the uracil and thiamine. DNA has thiamine, and instead of thiamine, RNA has uracil. Maybe that one we will play out as we proceed. So there are three differences: the sugar, the number of strands, and then the organic base. So I beg to proceed and describe transcription very fast so that we can be able to also look at translation. Now, what happens is that the genetic information stored in the DNA molecules in the nucleus, as we have seen, needs to be transferred to the ribosomes in the cytoplasm. How does that take place? So it takes place through an intermediary, and this intermediary is what we call the messenger RNA. Now, the messenger RNA is a single strand which is formed from the DNA strand, and the DNA strand serves as a template. So what happens in the process of transcription? Transcription is now that copying of the coded information in the DNA molecule onto the messenger RNA, so that the messenger RNA can move out of the nucleus and take that coded information to the ribosomes in the nucleus, I mean in the cytoplasm. So the double helix of the DNA will unzip, and of course, three nucleotide molecules will align themselves around the template. Maybe we'll be able to appreciate this as we look at some of the diagrams in the next slide. So the base sequence of the template strand, that is DNA, is copied onto the new strand of the messenger RNA, which then becomes an RNA strand. Maybe we can look at the next slide. I think what we have in this particular slide is basically what I have just described. Once the messenger RNA is formed, goes out of the nucleus with all the coded information or instructions. Then, remember what we said in our some few minutes ago about the triplets or the codons on the messenger RNA. Hope you can see that. So that coded information in the messenger RNA, once interpreted by the ribosomes will synthesize particular proteins, and the proteins produced will give the characteristics to the organism. So today you have normal skin color. You inherited a normal gene from your parent. That normal gene you inherited is coding for synthesis of melanin. That's why you have that characteristic. Now, the process of making proteins can be split into two parts. Transcription, it means the making of RNA. Then translation is now that process of making proteins, following the instructions that are carried on the messenger RNA. So that's why I said we need to understand the three terms: replication, transcription, and the translation. Now, what we have here on this particular slide the slide demonstrates transcription, the messenger RNA being formed from the DNA strand. You can see. So a single strand is produced. Now, this single strand has coded information in it. Maybe I can illustrate more. Where is my pen? I can illustrate more. Now, what happens is that I'm going to use red color to represent one of the DNA strands. Then I put the organic bases. Maybe there is thymine there, there is guanine, cytosine, there is adenine there, maybe there is guanine there. Now, let me pick a different color. Now, when it comes to the formation of this single strand of the messenger RNA, we've said that the DNA strand is used as a template. So, thymine, what do we have on this side? Somebody, we have adenine. We have adenine there. Cytosine, guanine pairs with cytosine. Cytosine pairs with guanine. This is adenine. Now, do we have uracil here or thymine? We have uracil. Here, we have cytosine. Then, this DNA, this messenger RNA is a single strand structure, and is the one that we are seeing here. I have drawn just a small portion. It is the one now going out with this coded information, and it goes out of the nucleus through the nuclear pore, so that in the cytoplasm where we have these ribosomes, they can be able to receive that coded information and synthesize the protein. So I think now from this slide we have the DNA combined. DNA molecule. This one is a DNA molecule consisting of gene 1. There is gene 2, gene 3. Okay. Then DNA strand. This one. The coded information being copied onto a single strand of messenger RNA. This is what we call transcription. Then remember the translation, the making of proteins, the positioning of amino acids along a polypeptide chain, which is determined by the codons. You can see, like this codon code for this amino acid. So we have a protein consisting of 1, 2, 3, 4 different amino acid molecules. So I hope it's clear, unless there is somebody with a question. Maybe we can have this one.
In order for our bodies to function, we need to supply them with a variety of nutrients we get from our diet. Our bodies cannot use the food as it is when it enters our digestive system. The process of chemical digestion uses different proteins and enzymes to break down the food particles into usable nutrients our cells can absorb. And where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes: transcription and translation. In eukaryotic cells, transcription occurs in the nucleus. where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliciosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation.
Very good.
How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids for RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down 1 codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and is again at initiation. The purpose of translation is to produce polypeptides quickly and accurately. After dissociation, the polypeptide may need to be modified before it is ready to function. Modifications take place in different organelles for different proteins. In order for a digestive enzyme to be secreted into the stomach or intestines, the polypeptide is translated into the endoplasmic reticulum, modified as it passes through the Golgi, then secreted using a vesicle
Okay, so I hope it was a wonderful video which has explained very well what I was trying to explain about the DNA forming the messenger RNA and the entire process of protein synthesis. Now, I hope it's clear, unless maybe there are questions. Unless there are questions. John Gechongo, can you hear me? John? John, can you hear me?
Yes, he's able to
hear you.
Okay, any questions so far? You know, this is supposed to be interactive. I'm happy when I get questions so that I'm able to know whether we are sailing in the same boat. So I hope where you sit, you now appreciate the role of DNA, the three roles of DNA: storage of genetic information, transfer of genetic information unchanged, and also that process of formation of proteins that determines the characteristics of organisms. So the reason why I may have taken a lot of time in trying to explain what a gene is for you to be able to understand the inheritance, the transmission of characteristics from from the parent to the offspring. You need first of all to understand the structure and function of the gene, so that as we look at the roles of heredity, as we look at inheritance of different types of characteristics and so on, at least basic you have the basics. You know what is a gene. Basic gene. You know that the functional unit of the gene. I mean the functional unit of the chromosome, the gene, whose function is to determine the characteristics through protein synthesis. Time seems not to be on our side. I beg to stop unless there are questions. Mr. Ahmed, I hope you can hear me.
Yes, Mwalimu, I can hear you. I can also see there are some students who had some questions, but due to lack of time, as the Form 3 class is supposed to start by now, I will just add you in the group, in their WhatsApp group tonight, so that you can find some time to answer some of their questions. That's directed to Malik Mukhtar, who has some questions here.
Okay, and if possible,
yes,
if you can also share their emails. I'm assuming they have emails.
Yes, they have a Google Classroom, and I will invite you, and I will add you to that Google Classroom by.
Yes, so that I can be able to give some assignment.
Thank you so much, Mwalimu.
I hope they will be ready to do the assignments and turn in on time, so that at least I can also get to know whether I'm having an impact on what we are doing.
True.
Otherwise, thank you so much for your participation. I want to believe you have enjoyed the lesson.